Thank you for joining me today. My presentation is all about color magnitude diagrams in open clusters. Let's begin. In this presentation, we will cover what is a color magnitude diagram, what is an open cluster, how it was done, and why it matters. To understand color magnitude diagrams, we first ask, how does starlight work? We study it in a few crucial ways, mainly by measuring how bright it is and what color it is. Comparing the brightness of each color reveals a lot about a star. We can get into that when we further ask, what does each of these tell us about? Brightness, henceforth called magnitude, tells us the power output of a star and gives us an idea of its luminosity. It varies with distance and isn't always one-to-one with temperature, so brightness alone tells us little about a star's composition. Similarly, a single color doesn't tell us much, but isolating brightness in multiple colors tells us a lot: temperature, age, and distance. From that, we can infer composition, metal fraction, and reddening, which we'll explain later. These are typically collected in UBVRI bands: ultraviolet, blue, visual, which is a yellow-green, red, and infrared. A Hertzsprung-Russell diagram plots spectral class versus magnitude. A color magnitude diagram is a real data version that plots color index against brightness, such as red minus infrared in the plot at right. They look very similar, so you may be wondering how they differ in practice. How do they differ? As shown in the table, an HR diagram uses spectral class, temperature, or B minus V color index plotted against luminosity. A color magnitude diagram, or CMD, always uses a color index against an observed magnitude. HR diagrams are harder to implement because they require absolute magnitudes or luminosity, which means you need to know the exact distance to a star. CMDs are much easier to apply, and they're one of the key tools we use to study stars in practice. An HR diagram typically uses spectroscopy, while a CMD uses photometry with a camera. Photometry involves drawing circles around stars in an image and summing the pixel values to measure brightness. A star's image profile comes from the PSF, or point spread function. This describes how a point source disperses at perfect focus. You can't collect 100% of a star's light without summing every pixel. It gets exponentially dimmer as you move outwards. In practice, you usually capture about 95% of the light. But, since the PSF is consistent across stars, this works fine. Brighter stars appear larger because their dim outer wings are brighter and more visible. Now that we've covered CMDs and photometry, what is an open cluster? It's a group of a few dozen to a few thousand stars, usually a few hundred, spread over about 30 light years. These stars form from the same molecular cloud at roughly the same time. Since they share age and distance and starting conditions, open clusters are ideal for benchmarking stellar evolution and testing models. Now we can get into the data collected. I used the Gamma Ray Optical Robotic Telescope, or GORT, at Sonoma State to capture two star fields through BVRI filters. One field was Messier 29, a young open cluster in Cygnus, estimated to be 10 to 20 million years old. The second field was a Landolt Standard Field, a set of stars with precisely known magnitudes in UBVRI. Observing them lets me calibrate my measurements to match established systems, making my CMDs accurate and comparable to other studies. Once we have the data, we measure each star's brightness in every filter using aperture photometry. Then we use a program called TopCat to match these stars between filters based on their coordinates, ensuring when we compare colors we're looking at the same stars. When we calibrate the data using our field of Landolt standard stars, we first correct for atmospheric extinction, basically how much the atmosphere dims the starlight, and then determine 0 points that turn our raw magnitudes into real ones. Atmospheric extinction acts unevenly across bands, which is why during the day the sky is blue, but at sunset it's pink or red. The light is passing through more atmosphere and getting biased towards the red end. The 0 points are essentially how far off your aperture photometry magnitudes are from the standard magnitudes, which is expected because every telescope and camera is different. Finally, we plot color versus magnitude to create the color magnitude diagram. Now the fun part. It's time for our conclusions. These are the three color magnitude plots I created with data from GORT. You can see that they are only in the V, R, and I bands without the blue band. This is because stars are immensely brighter in the longer, redder wavelengths. This means that due to GORT only being able to track accurately for 30-second exposures, the blue data was too noisy to get results. In fact, you can see that even the visual band data only has around 25 identifiable stars. This is because main sequence stars are an entire magnitude, 2.5 times dimmer than the visual band than they are in the infrared. The spread from our brightest star to our dimmest star is from about 8th to 16th magnitude. Since magnitudes are logarithmic, with 5 magnitudes being 100 times dimmer, this jump means our dimmest stars are 1,585 times dimmer than our brightest ones. This is why the detection limit for visual band stars was around 14th magnitude, while for infrared stars it was 16th, a difference of 6.3 times. I'll get into how to read them on the next slide. In these plots, the blue dots are stars confirmed as cluster members by Gaia parallax data. Gaia is a space telescope that measures the motion and parallax of stars to determine their distances. This is especially useful for clusters like M29, where interstellar reddening makes main sequence fitting difficult. On the right, you can see the large spread in parallax, with some stars having even larger error bars. I filtered for stars within the distance range, shown by the white x-axis line, and with small parallax errors. Stars are colored by R-I index, showing many bluer younger stars, and that most outliers are redder, consistent with M29's young age of 10 to 20 million years. The orange cloud beneath the points is an isochrone, a model population of stars formed at the same time but with different masses. By adjusting parameters like age, metallicity, and extinction, we can fit the isochrone to our data, revealing the cluster's properties. The isochrone includes a full red giant branch, unlike M29, because high-mass stars evolve quickly but are rare. It also extends to fainter magnitudes beyond Goert's reach, down to low-mass stars and brown dwarfs, though we can still study some dim main sequence stars with other methods. Gaia doesn't just measure parallax and motion, it's also a powerful photometric instrument. Here we've plotted an isochrone with the same initial conditions against Gaia's blue minus green photometry. Gaia's system differs slightly from Goert's, but produces the same type of plot. This dataset includes more confirmed cluster members, selected the same way as before but without requiring a Goert match, so the sample is larger. The fit to the isochrone is tighter and extends down to about 20th magnitude. Because Gaia's data is trusted by scientists worldwide, the fact that our own measurements match the same isochrone is strong validation. It shows that we determined M29's physical parameters using a relatively small amateur purchasable telescope and a 20-year-old CCD, and did it with just 2 hours of data taken on some of the shortest nights of the year between bouts of heavy fog. So what were those parameters? Based on the isochrone, the best fit for M29 is a very young cluster of about 20 million years, too young to have developed a red giant branch. It lies roughly 5,240 light-years away, based on the best fit distance modulus, which describes how much dimmer an object appears at a given distance. Its metal content is Z equals 0.014, or about 1.4% metals, slightly heavier than the Sun's 1.2%, Z equals 0.0122, with metals in astronomy meaning any element heavier than hydrogen or helium, such as carbon, neon, and iron. The cluster's reddening is AV equals 3.24, meaning 3.24 magnitudes of extinction in the visual band, which is intense, but consistent with the patchy dust in the constellation Cygnus, where M29 resides. Finally, its stars range from about 11.2 solar masses to 0.748 solar masses, with the most massive stars being about 11 times heavier than the Sun, and the smallest about 75% of the Sun's mass. Thank you very much for listening. I've been August Norman, and this presentation was only possible with the help of my mentor, Dr. McLinn, as well as the equipment provided by Sonoma State University and funding received from NASA and their partners. I will now take 2 minutes of questions about the project.